Hello, and welcome to this lecture on Parkinson's disease research. This is actually the second of two lectures, and ideally you should watch the first lecture before you watch this one. That was about an introduction to the science of Parkinson's. And what we're going to cover in this lecture is current research and emerging therapies, and we'll also take a bit of a look into the future and ask the question, when will we ultimately cure Parkinson's disease? I would stress that these are very much my own opinions, and if you speak to other people in the medical community, they may have different opinions. So there's an awful lot going on. If you look at the list on the left, and these are just some examples of some of the areas of research that are being carried out by the organisations on the right. But this is just a sample. There's a lot more that's not on this list. And moreover, I'm not going to talk through everything on this list. I'm going to focus on 3 or 4 items and some of the key things I think that are important in research going forwards. Before we get into therapies, we should talk about strategies for dealing with Parkinson's. So first of all, ideally what we'd all like is to find a cure. So I would like to take a pill that will make this condition go away. But if we can't do that, perhaps the next best thing is to talk about disease modification. So can we slow down the progression of the disease? Could we stop it progressing altogether? Or could we even reverse progression even just a little bit? So not cure it, but make it a lot better than it was. Another strategy is to look at, can we replace or regenerate some of the cells that are lost in Parkinson's? So we're not actually slowing down or stopping the underlying disease process itself, but we're just kind of patching up the damage, and that can actually achieve quite a lot. We can look at genetic variants of Parkinson's and stop them being passed on to future generations. And also we can look at better symptomatic treatment. So the best example of symptomatic treatment, of course, is levodopa, but that just treats the lack of dopamine. It compensates for the lack of dopamine in the brain. But if we could find other treatments like that that perhaps deal with some of the non-motor symptoms, that would be a big breakthrough as well. And finally, if we can't do any of those things, we can still look at lifestyle changes, in particular diet and exercise. So we'll talk a bit about that a bit later. So the key point here is that patients don't necessarily need a cure, although we'd all like to have a cure. We just need therapies that can give us a better quality of life for long enough. So let's look at the first of the items that was on the list a couple of slides ago, drug repurposing. If we look at the drug development pipeline, it goes through a number of stages, and each of those stages takes 2 or 3 years. So in total, from having a good idea in the lab, it can take perhaps 12, maybe even 15 years to get that out to ultimately being approved for use in patients. And all those stages are important. They look at safety, they look at dosage, they look at, of course, the efficacy, the effectiveness of the drug as well. But what we could do is if we could identify some drugs that are already approved for use in humans, we can cut out a couple of those stages. And that sounds fanciful, but actually a lot of work's been done on this. A couple of examples that are in current testing are Ambroxol, which is a cough medicine, and exenatide, which is used to treat diabetes. These have been led out of Cure Parkinson's in the UK, but there are other examples in other countries as well. And the key point is that because these are already licensed for use in humans, perhaps at lower dosages, the safety testing has already been done, so we can move straight to, in some cases, a stage 2, and we can get something out to market in perhaps 5 years rather than 12 years. So talking about exenatide a bit further, this is an example of what's called a GLP-1 agonist. And where this comes from is it's currently used to treat type 2 diabetes. And what we find is that people with type 2 diabetes are more likely to get Parkinson's. Moreover, if they're already taking what's called a GLP-1 agonist, they have a reduced risk of developing Parkinson's. So this implies that there's some kind of mechanism that's shared between the two conditions, and some kind of common factor. So obviously what we can do is target GLP-1 agonists for clinical trials in Parkinson's. That's been done over the last few years, and the most promising drugs are exenatide and lixisenatide, which both had very good results from phase 2 studies. Recently, a phase 3 study finished a couple of months ago for exenatide, and I think everyone was very much expecting this to be a breakthrough and be a drug that could be used to slow down the progression of Parkinson's. Unfortunately, it didn't meet its endpoints. What they found is that although there was some improvement in the patients that took exenatide, those weren't any better than the placebo patients, the patients taking kind of a dummy tablet. So that was obviously very disappointing. It's not quite the end of the road for exenatide, but we need to look closely at the data to understand why it had such promising results in phase 2 that didn't translate to phase 3. Lixisenatide is still under investigation for phase 3. So we're still looking for that first disease-modifying therapy for Parkinson's disease. So moving now to another strategy, can we replace or repair dopaminergic cells? So these are the cells that are getting lost in Parkinson's disease in this region we call the substantia nigra in the midbrain. One thing we can do is inject neurotrophic factors. So there was some work done a few years ago that looked at a chemical called GDNF, which is kind of like a growth stimulant for neurons. It encourages damaged neurons to regenerate and stimulates growth of existing neurons. It can't bring dead neurons back to life, but it can prolong the life of neurons that perhaps are damaged. And there was some quite promising results on that, but again, one of the problems was the placebo performed almost as well as the active arm. But nevertheless, there are going to be further studies on this starting up this year, and the results in the lab certainly look very promising, that there's definitely something going on, but what we need to do is figure out how to make it as effective as possible. So there's a new study that you can view in one of the other videos about GDNF, and we'll put a link to that in the description for this video. Similarly, we've also done a video on stem cell treatment, which looks very promising. I interviewed Professor Roger Barker at the University of Cambridge a few months ago, and we'll put a link to that video in the description as well. But stem cells are looking very promising. A number of big pharma companies are investing heavily in this, and there's at least 3 pharma companies interested around the world, and there are several clinical trials ongoing. This is to some extent proven. Experiments were done as far back as the 80s, with the general concept of putting a dopaminergic cell, a replacement cell, into the substantia nigra has been tested and did actually work in a number of patients. But there's a lot more work to do to understand why it works in some patients and not others, and how we make it effective and repeatable and reliable and safe going forwards. But personally, I think this looks very promising. I think we'll find out over the next couple of years whether these trials worked, and hopefully this will lead to a new therapy for Parkinson's. The third item on the list is reprogramming glial cells. So glial cells are kind of helper or worker cells in the brain that work alongside the neurons. And this is at its early stages, so it isn't something that's going to come to fruition in the near term, but it might be something in the longer term that's a viable option. As I said, we have a separate video on stem cells, but just in summary, the historical approach was to use embryonic stem cells. That was both expensive, you needed several embryos from aborted fetuses, and it was ethically challenging as well. The modern approach is to use what we call induced pluripotent stem cells, and you can see the picture on the right of the Japanese researcher who got a Nobel Prize for discovering that process. And this is basically where you can take an existing adult cell in the body and you can effectively reprogram it to become any other cell type, like a dopaminergic cell type, that can then be implanted back into the body or the brain. There are a number of surgical techniques for Parkinson's that have been used historically. So, for example, Michael J. Fox had a thalalotomy many years ago. It turns out that if you lesion, that is, if you remove some very small parts of the midbrain, you can actually alleviate some of the symptoms of Parkinson's. So, for people who have very severe tremor, you can take out a piece of the brain called the motor nucleus of the thalamus. And if you have severe slowness of movement and rigidity, if you take out the subthalamic nucleus, similar name but different part of the brain, then you can also dramatically improve that as well. I actually went to see this procedure being performed in a hospital in London, and it's really quite remarkable what it does. I saw people coming in with essential tremor rather than Parkinson's tremor, but it's effectively the same thing. And they were shaking really very violently. They were unable to function normally. Within half an hour, they were wide awake, and they could start writing these spiral tests where you draw around a spiral really quite fluently. And it's remarkable to see the transformation within 30 minutes, as I say, of someone who's wide awake during the whole procedure and really doesn't feel very much going on. And the way this works is ultrasound beams are sent in all around the scalp in a very mild dosage, but they focus in on the central point, and at that central point it's hot enough that they can actually lesion the part of the brain. So I think this is a new surgical technique that is a lot less invasive than traditional surgery. So this is complementary to DBS, deep brain stimulation, which is a slightly different technique where you implant electrodes into the basal ganglia, into the midbrain, and you send electrical signals down. The advantage of DBS, although it's a surgical procedure, the advantage is you can modify it. You can change the nature of the electrical signals, or you can even switch it off if you like. So it's much more adaptive. Whereas focused ultrasound is a kind of one-off procedure that you can't reverse. And there are also many other innovative technology solutions being developed. At the last World Parkinson Congress, I saw a number of these, and we've actually made videos out of several of these, which you can view separately. Again, I'll put the links to those in the description of this video. But there's a lot of clever ideas about how you can, for example, counteract tremor, and how you can help people overcome their freezing and so on. Finally, let's talk about lifestyle. So there are two key elements here. There's exercise and nutrition. There's also stress, which it seems to be a contributory factor in symptoms getting worse. So if you can remove stress from your life, that's undoubtedly a good thing. But if we talk about exercise first of all, this is the one thing that's clinically demonstrated to slow progression of the disease. Specifically, high intensity cardio exercise is the most effective. So something that increases your heart rate to a reasonable level. But if you can't do that, then any type of exercise is better than no exercise. The benefit is cumulative through the day. So ideally, you do an hour's exercise in the morning or whatever, but it doesn't really matter if you break that up into 5 or 10 minute chunks throughout the day. They all have a benefit. So keeping exercises is the key, and finding an exercise program that works for you. I run and bike, but equally you could do yoga, you could do shadow boxing, you could do a dancing class, even just walking upstairs, doing DIY or gardening is all beneficial. So do try very hard to exercise as much as possible. It's the one thing that you can do for yourself that will improve your outlook with Parkinson's. It won't necessarily stop you getting Parkinson's, or maybe delay the onset of Parkinson's, but once you've got Parkinson's, it definitely slows down progression. Dietary impact on Parkinson's is less clear cut. I think there's a lot of anecdotal evidence, there's a lot of people talking about different things that work for them, but there's less scientific evidence that something definitely can make a difference. But nevertheless, the way I look at this is in three levels. So level one I think of as a general healthy diet. And I think everyone who has Parkinson's should be doing this anyway. So cut down on the red meat, cut down on the processed foods, eat as organic as much as possible, keep hydrated and eat lots of fresh fruit and veg. And that will also help with constipation, that's a common symptom in Parkinson's. So I would certainly encourage everyone to do that kind of healthy diet or Mediterranean style diet. As I say, there's a lot of research going on into what specific things can you do that are good or bad for Parkinson's disease. So Dr. Laurie Mishley in the US has pioneered this field. She's taken a large number of patients and she looked at which of them are doing well and which of them are doing less well in their Parkinson's progression, and what are they eating or not eating or drinking. So she's found things like dairy is kind of bad for Parkinson's, coconut oil is good for Parkinson's. The dairy thing I've heard from several places, and the theory is that it interferes with the uptake of levodopa because the same enzymes in the stomach that process cheese and milk, for example, would also be processing the levodopa. So if you eat dairy, particularly around when you take your medication, you'll have much less effect from the medication. And I must say, anecdotally, I kind of observe this as well. I certainly notice that when I eat and take my medication at the same time, the medication has less effect, and when I have dairy specifically, it does seem to be significantly worse. So I think there's probably something in that, but as I say, there's less solid scientific evidence for this. The third level, which is getting quite a lot of airtime at the moment, is ketogenic diets. So this is the no carbohydrate diets. And similar to these are fasting diets where you go for half a day, or even a couple of days without eating, just drinking water. And what that encourages in the brain and the body generally is for the mitochondria, the energy producing factories in cells, to burn fat rather than sugar. And that burning fat is a healthier process that allows the cells to sort of recover a little bit and does less damage to them. There's a doctor called Matthew Phillips based out of New Zealand who's done quite a bit of work on this. There's some links at the bottom of this slide to various YouTube videos where you can find out more about this. But again, the same issue is that it's certainly a promising area. It certainly makes sense from a logical perspective, but whether there's genuine improvement to be had for people who already have Parkinson's by doing these extreme diets is less clear cut. So finally, when will we actually cure Parkinson's, or at least have better therapies? So if we look at the amount of research going on in Parkinson's, I did a simple search on a database called PubMed, which is one of the main sites for all the publications in medical research. And I searched for the word Parkinson in the title. If you do that, you'll find about 5,000 papers a year currently published. And that's a number that's increased quite dramatically over the last 20 years or so. So that's good news that research output and by extension research funding is going up quite steadily. But if you compare Parkinson's in the graph on the right to other conditions like HIV/AIDS, diabetes and COVID, it's still quite a long way behind. If we look at the research process, the funding comes from really two sources, governments and charities, but also pharma companies. What governments and charities do is give money to universities to fund individual projects. Those projects are either fundamental research, which results in papers being published, or they might be to develop therapies. Pharma companies will also develop therapies. They're less likely to do fundamental research, although they may occasionally do that. Obviously, therapies result in clinical trials, and as a side effect might publish more papers. And ultimately, what we're looking for is a new therapy to get produced as part of all this investment. So if we look at Parkinson's specifically, there's some problems with this, and one problem is the animal models that are used to test drugs in Parkinson's, like MPTP, are really not very effective. So MPTP is a type of heroin actually that's injected into the brains of mice. Again, there are ethical challenges with this, but anyway, it's injected into the brains of mice and it induces a kind of rapid form of Parkinson's. And when you give certain drugs to those same mice, like levodopa, it has a dramatic effect in reversing their symptoms. So this MPTP model and a couple of other models have been used for many years to test prospective drugs. But what we found every time is something that works on MPTP doesn't translate to humans. So I think it's questionable whether this model that brings very rapid Parkinson's is actually very useful in trying to mirror a disease which is very slowly progressing in humans. We've already talked about clinical trials, expensive and take a long time. Current research is quite piecemeal, and data is not always shared as well. The other thing we need is more fundamental science. We're shooting in the dark a little bit when we try some of these drugs. That said, medical science is littered with many examples of serendipitous discoveries. So, for example, people discovered that eating fresh fruit could stave off scurvy long before vitamin C was discovered. So we might get lucky and discover some new drugs that work with Parkinson's, but the more fundamental research we do to understand the underlying biochemistry can only be a good thing. So another way of improving clinical trials as well as drug repurposing is this idea of MAMS, which is multiple arm, multiple stage trials. And the idea here is that you, rather than setting up a whole new trial every time you want to test a drug and then running the test, having a placebo arm, and then winding down the trial and doing the analysis, it's a very inefficient process. Is you could set up kind of a factory or a machine that just continually runs clinical trials. So the idea is that you have a single placebo arm and multiple trial arms, so that's already more efficient and cheaper. You're only using one placebo and several drugs in parallel. And then you evaluate these things perhaps every year, and you replace the ones that aren't working with ones that new ones that have potential. And so you keep this pipeline continuously going. You feed new drugs in all the time, and if something is looking promising, then you see it through to the end of this 3 years or however long the phase 3 trial needs to take, and gather the data for that. So there's an initiative in the UK starting up this year called EJS, Edmund J. Safra, ACT PD, Accelerating Clinical Trials in Parkinson's. So do look out for that. So this guy is Sergey Brin. He's one of the co-founders of Google. He's one of the richest people in the world, but he also carries a genetic variant that can cause Parkinson's, the LRRK2 variant, and his mother has that variant as well, and she already has Parkinson's, but he hasn't yet developed the disease himself. He probably has a 50-50 chance of getting Parkinson's. But what he's done is he's used quite a bit of his wealth for the benefit of Parkinson's research, and in particular he put a couple $100 million into launching something called ASAP, Aligning Science Across Parkinson's, which is focused on fundamental science, and it has principles of things like collaboration and transparency. So the idea is that people get funding, but only if they're willing to work together and share their data and share their ideas. And so the idea is, as I say, to focus on the fundamental science to really understand what's going on with Parkinson's. So if we put all these things together, I think it's fair to say we're doing all the right things. We're improving the clinical trial process, we're repurposing drugs, we've got the MAMS trial starting up this year in the UK. We're doing things like better endpoint measurement, so better tracking of the symptoms of Parkinson's using, for example, wearable technology so that we can more accurately and more consistently measure how effective a particular therapy is. The ASAP initiative, which I just went through, improves the funding model and focuses on basic science and encourages data sharing. We're starting to understand the variation of Parkinson's better, and to answer this question of are there actually different subtypes of Parkinson's. Genetics is very active, and there are also these alternatives to curing Parkinson's or disease modification, which around cell therapy and other alternatives. And there's a steadily increasing awareness of PD and overall investment going into research. So let's bring this all together. This is my crystal ball. Again, I would stress this is a personal view. Other people may have very different opinions. I think it may sound slightly pessimistic, but I think it's also a realistic view. And as I say, there are some things to be positive about. So over the next 10 years, I think we'll see some new drugs that will slow progression for some people. There are many drugs in clinical trials. I think it's highly likely that one or two of them will come good. I think focused ultrasound will be a mature and viable alternative to surgical techniques. And I think we'll start to see more personalised symptomatic relief, and perhaps supplemented by some of these technology solutions that we talked about earlier. Over the next 20 years, I'm fairly optimistic that cell regeneration or cell replacement will come good, and we'll have maybe one or two new options to not slow down or modify the disease progression, but actually to repair some of the damage that it's done. In terms of actually fundamentally curing Parkinson's and really slowing down, halting or reversing the underlying condition, I think we're probably 20 or 30 years away from understanding the genetic forms of Parkinson's. And in terms of idiopathic Parkinson's and ultimately, curing all the cases of Parkinson's, I think that's a generational problem. I think there's 30 or 40 years work to do before we can really get rid of Parkinson's. But I guess the key point I'm making is, although we might be a long time away from really ultimately curing the condition, there's an awful lot of things that I think will come good in the meantime. So thank you very much for listening. Do please post your comments on the video, and I hope you found this useful.